Today, we're very pleased to welcome Marjan Slak Rupnik, PhD. He's coming to us from the Center for Physiology and Pharmacology at the Medical University of Vienna. The title of his talk is Intracellular Calcium Flux Interactions in Beta Cells. And just a little bit about him and his background, a little short bio here in the PhD in cell physiology from the University of Ljubljana. He was trained in electro and optophysiology using techniques such as patch clamp and single multiphoton confocal imaging, and following numerous international research stays at St. Andrews, Cambridge, Wisconsin, Paris, and Tokyo. He moved to Göttingen, Germany, where he took on the role of junior group leader at the European Neuroscience Institute at Max Planck Institute for Biophysical Chemistry, and his main aim was to establish several endocrine slice preparations, including pancreatic tissue slices. In 2005, he was appointed as the founding chair of the Institute of Physiology at the Medical Faculty of the University of Maribor in Slovenia. And since 2014, he's led a laboratory at the Medical College of Vienna, Austria, focusing on cytosolic calcium homeostasis and beta cell collective activity. The lab uses advanced imaging and analytical approaches, and his research has been supported by grants from the Max Planck Society, the Austrian Science Fund, the Slovenian Research Agency, and Vienna Science and Technology Fund, and the NIH, along with Carmela Evans-Molina at Indiana University. Welcome, Marjan. Thank you very much for joining us today. We had a great discussion at the ADA, and I'm very pleased that you're coming to join us live today to discuss your work.
Thank you very much, Monica, for inviting me to present for the Sugar Science. Many years ago, I was shortly running a blog called Why Only Glucose? Then I just got too busy with stuff because I always thought that there is, things might be a bit more complex than just sugar. But so I will talk a little about that today, but of course, we only started to actually explore what is actually going on. With the next 20 slides, I would like to share with you our experience for basically what we did with slices for the last 20 years and how what a hardcore physiologist could learn. And I'm still constantly learning from these experiments on the slices. And I'm really happy to tell you that it's getting more and more interesting with every year, which is very exciting. So today I would like to expand the consensus view, particularly regarding the calcium homeostasis and calcium flux in beta cells, particularly regarding its sources and operating speed. This is two things which are actually, I think they are not really fully covered with the mainstream research on that. We started chasing intracellular calcium release about 20 years ago, but really made some progress only recently in the last 5 years due to technological development, and also because we could connect some dots with discussions with our dear friends, basically where we actually could see why we were not able to actually see more about this in intracellular calcium fluxes before. So before I run out of time, I would like to present the people who actually did the work, major collaboration, and also the funding, which actually made our work possible. So we started slices at the Max Planck Institute for Biophysical Chemistry in year 2001. I will, in the next slide, tell a bit more. But then we moved the technology to University of Maribor and then also to University of Vienna, where several very talented people like Sandra, Johannes, Nastya, but also in Maribor, Yasmina, Lydia, and others actually could really work with this preparation and actually do really wonderful things with that. So because it's a very complex system, so I always searched help from physicists, data scientists that actually enabled us biologists to understand a bit better, these complex interactions between the cells, because we were basically not trained for that during our basic education. True boost and with access to the resources of intracellular calcium released came when we started to NIH grant together with Carmela Evans-Molina and Jim Johnson from Indianapolis and Vancouver, respectively, as experts on intracellular calcium release. Actually, they could really share with us some very vital information that also changed the way how we actually started doing experiments and actually also do analysis. I would also like to acknowledge the work of Yachi Huang, who is now in Montreal, and very nice discussion with Wolfgang Greyer and Thomas Pieber from Medical University of Graz, which actually gave us some critical hints what we could change to actually make it work better. Because our data is very data intensive, you know. So we recently got Vienna Science and Technology Fund together with Gaspard Kacik from Institute of Science and Technology in Klosterneuburg near Vienna, where we actually hope to basically use tools of machine learning for beta cell biology and basically disentangle the patterns of intracellular calcium release and understand collective activity of beta cells much better. I will mostly talk about mouse data today because I believe that for humans, we don't use proper stimulation protocols yet, and we cannot really get all this, the bright spectrum of activities that we actually can get from the mouse. But still, I mean, as we get more preparations from donors, actually, we will do it in the future much, much better. So what we're gonna hear today, I will first use 2/3 of my talk to actually talk about some special features of beta cells or islets in pancreatic tissue slices, since as many things are not really well understood in the field, and often people use it with some kind of like prejudice, which may not be completely true. And then I'm gonna also introduce high throughput analysis of large databases that we actually had to establish to go through our data. I will present how we stumbled upon intracellular calcium release, particularly interplay between IP3 receptors and ryanodine receptors. And actually that led us to think of ER as some sort of interactive fountain. So this is the Hochstrahlbrunnen in Vienna, which is a symbol of us drinking very, very nice alpine water in Vienna here. But it's just an example that you can have different amplitudes of different jets, have kind of small jets or can high jets, but this fountain is very interactive. If you close one, you can actually influence the other, and there are some interaction also between the IP3 receptors and ryanodine receptors in beta cells. And then I will finish with just some information about the stimulation paradigms that we're using and how multidimensional actually stimulation can be. It's not just low glucose, high glucose, but it can be many, many different layers between that, which actually give us critical information about what is going on within beta cells. The whole thing started about 23 years ago in Göttingen. This is me 23 years ago, where Erwin Neer approached me because I was still working mostly as a patch clamper, so that's why it was okay, that I was working close to Erwin Neer in Göttingen. They had an issue, and they wanted to acquire a European grant, but the reviewer asked them likely to block the grant if they can do slice preparation of pancreas. So this is Klaas Wolheim, Fran Ashcroft, and Patrick Rossmann. And basically, my job was only to establish pancreatic slice because I was doing other endocrine tissue slices. And this would be like a difficult part of the job. And the easier part of the job would be to actually confirm everything what actually they showed on isolated cells before, so that they are KATP channels, which can be closed by ATP. And this is all glucose dependent. And then when the potassium channels close, this depolarizes the membrane, and the voltage activated calcium channel can activate and yield calcium then for extracytotic fusion of insulin granules. It turned out that basically slice was the easy part. We could not really confirm everything what actually was shown, and I will show you why actually this has been so. The first special feature of the slice preparation is that it has a completely different sensitivity. The KATP channels have completely different sensitivity to ATP than in more isolated preparations. So here I have on the left side top, I have everything what is related to the single cell. And when I would actually dialyze the cell with 0 ATP, I would wash out ATP, and that's why the KATP channels would open. I can see they are KATP potassium channels because they rotate around the reversal potential for potassium, which is defined by the ratio of concentration of potassium outside and inside. So when I apply more ATP, like 2 millimolar ATP, or if I block with it with sulfonylurea or butamide, very high concentration, basically, this is likely micromolar here, then I will actually block this potassium activity. But if I do the same experiment in beta cells within the slice, I can see that basically the 0 ATP opens my KTP channels much faster. And even at 2 millimolar ATP, not all of them are closed. And if I do the whole concentration dependence, I could see that basically how effective inhibition in the isolated cell is about few hundred micromolar, while in the beta cells within the slice, it is in the millimolar range. So practically, we don't need the concept of ADP, PIP2, or any oleoyl-CoA things that actually has been developed for the preparations which are more isolated, either isolated cells or even isolated channels. In the isolated cells, basically ATP-ADP concept doesn't work. Even if you have 1-to-1 ATP-ADP ratio, basically the KTP channels would not open. Yeah. They open only if I take these channels from this exactly experiment and I put in excised batch. So this is experiment below, so I can measure single channel activity. And then ADP, PIP2, or oleoyl-CoA would open the channels, but not in the context of whole cell, particularly not in the context of whole cell within the slice. It means that basically the ATP sensitivity and this ATP-ADP ratio and everything, this is a bit different in the kind of slice preparation. And the second feature of slices is that basically, because of this different sensitivity, there are other compounds, other parameters that actually influence the sensitivity, ATP sensitivity of KTP channels. So if I would dialyze NADH at concentration 1 millimolar into the cell, I would desensitize KTP channels to ATP. So the KTP channels will start to open. If I would increase calcium with, for example, acetylcholine stimulation of the M3 muscarinic receptors and actually releasing calcium from the intracellular stores, I would decrease the sensitivity to ATP, suggesting that basically, KTP channels could also be called calcium-dependent channels, so they could be called NADH-dependent channels. Every situation where calcium is high, we will also have more closed KTP channels, suggesting that basically, what is these causal correlations within the cells is not so clear on the level of the intact cells. So, and then in addition to ATP closing KTP channels, we can have other influences which desensitize or sensitize closure of the KTP channels when ATP is high. So what is then, we were chasing intracellular calcium release, as I said, for 20 years. Why we did not see anything? One of the major reasons is that we were actually trying to keep our cells inactive with glucose, which was too low. We used 3 millimolar glucose, and as Tenkholm already in 2001 showed, is basically the loading of organelles with calcium depends strictly in beta cells on glucose. So if you move your cells into 3 or lower glucose, you will actually deplete calcium from the ER. And then, of course, you cannot study calcium release from ER. So when we shifted, so basal glucose to 6 millimolar, which is also non-stimulatory in Black 6J strain mice, basically we could actually load calcium into the ER to the extent that we could then release it when we activated IP3 or ryanodine receptors. Maybe special feature about slices is also that we have this principle, what you see is what you can cut. And that you can basically, in addition to extracting beta cells or islets from the pancreas, we can also extract the context. Yeah. So in NOD mice, we just don't isolate islets, what we would do in islet isolation, but we also isolate the immune kind of context with that. So all these lymphocytes, macrophages, which actually attack beta cells, can be isolated together with the islet cells. And the second thing is what was very interesting, because in Göttingen at that time, we had lots of knockouts, which actually had perinatal mortality, and we wanted to study beta cell function with that mutants too. And we had to develop, so actually the slice preparation also to be able to work with perinatal preparations. And in perinatal preparations, we can easily see how we can extract the context. These are, with asterisks, I can always label this, the islets, but all these islets are connected with neurons, so basically they form also networks on the level of the whole pancreas, and we typically don't see that in adult preparation just simply because the distances are too big. What we also found in islets is that the structure of cells seems to be different from that in the isolated islets. So in isolated cells, the cells are kind of like fill a lot of space. They can be round or something like this, while in the beta cells within the slice, they have kind of like microarchitecture that very, very resembles the so-called Voronoi tessellation distribution. And Voronoi tessellation is a very known concept in techniques, particularly in communication technology, where you can actually plan the cell distribution for the mobile phone or something like this, actually doing this. So Voronoi tessellation suggests there is very efficient cell-to-cell communication, that the cell can very efficiently distribute resources. You can actually have spatial segregation and functional zoning due to that. There is a little signal interference. So all this tells us that basically, this arrangement that it is within the intact slice, within the intact islet within the slice, actually has kind of a deeper meaning than just, beta cells being together. And this is often forgotten when we think about it, when we try to compare isolated islets with the slice preparation. But there will be more differences, which I will list further on. And then we can actually kind of see a lot of different tissues within the slice. We can actually image all of them at the same time. We actually had to focus mostly on beta cells simply because, physiologies of different cells are very different. But we can very efficiently image that, particularly when we use low affinity calcium indicators, which I will also describe a little bit later. They give us very nice resolution when we stimulate them with 8-glucose. We will see that the cells completely change their activity when we add acetylcholine to stimulate M3 receptor, push them into the IP3 signaling, and then we can rescue this function with high sort of stimulatory ryanodine concentration later on. I will play the video now. There will be a rhythm that you will see how the cells actually, what kind of rhythmicity they have in 8-glucose, how they actually get very lost, non-coordinated with acetylcholine, and how high ryanodine actually can restore this function. Hopefully we can hear that. So at 8-glucose, we can see, how the islet actually, has these waves which can come from different sources, but there are some directions which might be conserved. Yeah. Then I switch to high acetylcholine now. Oops, sorry. It was just a little bit too much, too fast. Sorry. Let's start again. Might be the easiest. So this is the original rhythm first with high glucose. Cells are coordinated, particularly in the later plateau phase. We can see how this base component is completely gone with acetylcholine. The events are much faster, and then with ryanodine we can actually restore the function back. To analyze something like this, we had to develop the high throughput analysis pipeline, where we could actually correct the movement and define where the individual region of interests are. And then within those region of interest, we can actually, extract traces and then distill individual calcium events. Different time scales can be very fast, sub-second, can be about second long, can be hundreds of seconds long. We can define them all. And when we draw this experiment, then I actually showed you before as a raster. So each of the lines means individual cells. We have a bit more than 250 cells here. When we stimulate them with glucose, they seem to be very nicely coordinated, all of them active. But this rather high concentration of acetylcholine actually it stops the activity in some cells. They make them very fast activity. If I would do area under the curve, the cells here are definitely secreting much less insulin. And this is not the first time we observed that acetylcholine would inhibit the activity of beta cells. Already Sanchez, Andres, and Soria in '91 actually had a paper which was very controversial at the time, that acetylcholine can also inhibit insulin release. But as I said, when we had washout at the choline heteroiodine, we have kind of like smooth transition into kind of the rhythm that we actually had before.
Marjan, can I ask a kind of a side question here? Has
Yes.
anyone image slices in context of sort of a viral infection, like a CVB, added and then, take it away? And do you think, or if they have, would it mimic this acetylcholine event?
I don't know. I don't know. I don't know that anybody would actually. We were thinking about that, but it appeared to us that at the moment it was a bit more too complex for us to kind of like have all from viral infection to, because you need to change your lab a little bit to actually be able to work
Yeah.
with that.
And the
So
time points are different too, but
yeah, but definitely it's not acetylcholine. I will show you some vasopressin activity. Likely, if I would show you arginine stimulation, they mostly follow the same pattern. Yeah. So, but we
interesting.
were so far focusing mostly on the more physiological neurotransmitters and nutrients that actually could
Right.
be possible, but we
I
didn't
just
do
was curious if anyone had done that, but yeah, this is great, beautiful.
yeah. So how we analyze the data is that you can see an islet here with all the beta cells indicated or islet cells indicated. The cells in the center are a bit more active than the ones on the periphery. This we could regularly see. And when we see the activity, you see a lot of events. You have hundreds of thousands of events within maybe an hour or something like this. And each event has certain heights. And then we measure the duration at the half of the amplitude. We can also, correct for the different noise levels that you're bleaching. Instead of height, we can measure z-score or something like this. So there are several different features we can do. And we can see that basically we can have events which are some almost 100 seconds long, so very long events like that, more than a minute. We can have majority of them are few seconds long, but there are also quite a few which are sub-second here. And what is interesting is that basically most of the activity, we have a long event here, but this event actually has a lot of small events on top of that. But if you take one of these small events here and actually zoom in, we can see that each of those events is also made, kind of many, even shorter events, so that everything what we see here has a lot of self-similarity. It means that basically it's just summation of some individual parts, like I would say like some sparks from the ER release or something like this, And that basically beta cells are never really quiet. This would be 24 hours in the life of a pancreas, and you would have kind of like 3 meals per day. And each oscillation within this kind of meal, would be still kind of composed of smaller oscillations. So this is our pancreas beta cells, actually, they work very fast. It's just that what we typically measured are just temporal summation at a shorter time, kind of longer time scales that actually we were appreciating. We never measured really all this kind of like faster activity before. So event sorting and reproducibility. If I have two stimulations with 8 glucose within the same islet, so this would be the first stimulation and the second stimulation. The distribution of events, so the ultra short and long events, would be very, very comparable. Yeah, there might be differences between the mouse strains. So this is C black 6J in comparison to FVB, which we worked in Nashville. Our preparation are at the moment running in Indianapolis, Gainesville, Montreal, New York, Taipei, Gothenburg, and of course also Maribor. And basically, we can see that these strain differences, can be, the dominant component can be a bit longer or a bit shorter depending on the strain. And what you can clearly see here is that when I increase the glucose concentration from 8 to 12 glucose, you can see that the temporal summation gets more and the events get just longer, but lower frequency because of that. So you can clearly see how these peaks actually smear towards longer when I actually increase the glucose concentration in these mice when we did in Nashville. Yeah. So previously it was calcium, cytosolic calcium has not been really as a proxy, as a real proxy for the insulin release, because they sometimes people actually found that the two signals are kind of dissociated. So these are all data from Jean-Claude Onquin's lab, where they showed, for example, that when the cells are stimulated with 15 glucose and then epinephrine has been added at 1 micromolar, which is very high. Basically, you can see that the calcium, cytosolic calcium, actually did not get inhibited by epinephrine, but the insulin release has been. And we always thought that basically these are kind of like two processes are kind of dissociated, but what we realized very, very later on is basically that this likely is just the kind of like the side effect of using high affinity indicators like FURA2 or something like this. Because if you are, changing the concentration from here to here because you are lowering the calcium, the sensitivity of the indicator does not allow you to actually see the difference between this inhibition, when you inhibit your cells. But if you use low affinity indicator, like I will show you experiments a few slides later on, also with epinephrine, you are in this range here, you are much more in the linear range, and you can easily see like differences like almost 50% when you actually add epinephrine, suggesting that there is way more kind of like that the calcium is a really good proxy for actually insulin release. And also, we can also see differences in glucose concentration. Yeah? So when you apply 8, 9, or 12 glucose, you can see that you have several levels of temporal summation. It's something like tetanic contraction. So basically, if you stimulate your system more of these fast events actually fuse together and give you then higher and longer event. But basically, beta cells don't have to be stimulated that hard. In the slice, we can actually have physiological sensitivity to glucose. So this experiment here shows you that if I move glucose from 6.5 to 7, which is hardly believed to be high glucose in many experimental paradigms, I can actually fully activate my cells. And if I actually then do a more deeper analysis of that, I can see that activation of beta cells follows more or less so that the first order phase transition activation, which is kind of like which physicists know much more about that. We are not the first one actually to have measured that in more kind of in situ conditions. All these open squares here actually come from the publication in Journal of Physiology 95, when again Soria's group showed when they did sharp electrodes in the living mice, actually they could see that basically the cells were activating very, very fast. They did not interpret it at the time. They were actually still fitting the sigmoid curves through that, but their data actually fits perfectly to our data. Another feature is that basically we have physiological sensitivity, not just to glucose, but also to neurotransmitters like epinephrine in this case. So when I stimulate my cells with physiological glucose concentration, I can use physiological epinephrine concentrations to block the activity. And you can see that the amplitude of the events actually dramatically decreases before we actually completely lose the activity. So, the calcium, cytosolic calcium, is a very good measure of how much insulin I will release through that. I just have to have a more proper measurement of cytosolic calcium. It has to be low affinity to basically to detect that. So this is the concentration dependence of either alpha cell stimulation, which is also shown here. So when increased epinephrine concentration, I can actually activate alpha cells through activation of the beta adrenergic receptors, but I would inhibit the activity of beta cells through activation of the alpha-2 adrenergic receptors. It is also very interesting that alpha cells kind of stop before beta cells begin. So how much these cells are actually coupled in paracrine sense, we would have to really be more precise with trying to measure that. Another feature is that we are able to measure intracellular calcium release. Again, using comparing high affinity and low affinity experiments to each other is sometimes problematic. We have to imagine high affinity indicator is like a runner running through the mud, because it has high affinity to substrate, it cannot be very fast. And likely we are exaggerating the baseline oscillations, the baseline slow oscillations, but we are missing a lot of the fast oscillation on the top. And low affinity indicator means you're running on the hard ground, so your contact affinity is very low, so you can run faster. This experiment here is done with Fura. Actually tries to debunk our idea that there are RNA receptors in beta cells. But I would actually claim that, it really depends on the experiments. I can also find this kind of an experiment in our arsenal of experiments that actually shows that it takes quite a lot of time before high ryanodine would actually start working. And this is because we have several sources of calcium within the cells. We have IP3 receptors and have ryanodine receptors. And typically, IP3 receptors would activate first. So this would be the first part of activity, and this we would call first phase, which actually transits into the second phase, which is kind of different. The kind of events are a bit shorter, a bit more regular. When we wash it out with low 6-glucose and then activate it again in the higher ryanodine concentration, is basically the first phase, which was supposed to be IP3 receptors, is not really changed. But when we come to the point where the second phase should activate and the ryanodine receptor should take over, basically there's practically no activity. There is a huge rebound at the end, which we might not have time to actually go into that. And all the activity that is remaining here is likely the IP3 activity, because this kind of ryanodine inhibition should be irreversible. But they are both systems which are can run independently. So we actually suggested a bit more complex idea, not just 3 players on the plasma membrane, but that's also intracellular calcium release with IP3 receptors linked to many different GQ-coupled PLC-dependent signaling events driving IP3 release, or you would have ryanodine receptors. Basically, we also, we have to understand that KATP channels are not the only target for ATP. Circas, ryanodine receptors are direct, where ATP is a direct activator, direct ligand for them. Yeah. Then all these receptors, IP3 receptors, ryanodine receptors, can be post-translationally modulated. We actually tested that for protein kinase A. We have also some initial data for other post-translational modification within our NIH grant. So this is another layer of complexity which can actually be. We know from neuroscience since decades that basically often in neurons, IP3 receptors and ryanodine receptors would coexist in the cell, and then initially IP3 would activate because they have inactivation properties in the lower concentration of calcium, while ryanodine is like a safeguard. If you get really strongly stimulated, then you would activate ryanodine receptors in that conditions. And of course, I said there is interplay between those 2 receptors. So let me show you some more experimental evidence regarding this interplay, some more features about the differences between IP3 receptors and ryanodine receptors. This is a similar experiment that we watched a movie from. So we had first 8-glucose with the plateau activity. We pushed it into the IP3 mode with strong acetylcholine stimulation. And then even despite the fact that we actually removed acetylcholine, this activity, you can also see that the events get shorter and shorter, likely deplete ER of calcium. And then the cells, despite the fact that, maybe KATP channels can be closed and everything, it's enough energy still there. They need some time before they recover. We could see that basically if we added ryanodine right away, this kind of like rescue was much faster. Another feature which is interesting here is that, the cells are very nicely coordinated when we are in the ryanodine mode, what we call. So the mean degree, so we do a complex network analysis, is rather high, but when we apply acetylcholine, the network actually just goes away. It's not there anymore, and then basically recovers when we remove acetylcholine from the system. The second example is gain of function. So we have this mutant, we have a K channel. So this is TOC1 channel, which has a gain of function. So the cells cannot depolarize. And in this particular condition, 8-glucose is completely inefficient stimulating those cells to actually do calcium oscillations. But since they have IP3 receptors, they can completely equivalently to what we could see in the normal control, actually have acetylcholine response which has little coordination between the cells. So these two systems can basically work more or less independently. We can also play with blocking the the system closer to the receptor, to the GQ coupled receptor. We can actually couple, so inhibit the GQ protein separately. So in this experiment, I would first stimulate the cell with 8-glucose. It works, IP3 receptors work better if you actually support the activity with forskolin, low concentration of forskolin, which on its own does not actually activate beta cells. And under this condition, we used AVP, so arginine vasopressin, to activate their signaling pathways through the vasopressin V1B receptors, PLC, and then IP3 activation. And what happens here is basically we see there is a lot of activity in this sub-second range. So this is where we will completely miss that part if we don't measure with 20 Hertz or something like this, because it's just too fast. And then we apply the inhibitor of the GQ, G protein. We will see that we can, in concentration-dependent manner, inhibit that activity which was stimulated with vasopressin. So this peak here will be completely diminished with a progressively higher concentration of the YM254890, and we can also see here, yeah. And what is very, very interesting is also when we have a triple protocol here, and then we add the inhibitor, GQ inhibitor, just in the middle segment when we stimulate this triple glucose protocol, we can see clear first phase here with a plateau phase later on. We wash the system out. And then when we apply 8-glucose together with inhibited GQ signaling, basically, on gross, there is not much difference. But when we analyze the timing to activation, when we activate the density of the network, we see that this network here is 4 times stronger, 4 times denser than actually would be under normal conditions. So it means that if you block the IP3 receptors, the beta cells work more coordinated. If this is good or bad, this is another question, but this is what actually happens if you actually block the IP3, the GQ IP3 signaling. This is a textbook figure from German medical physiology textbooks where AVP is listed as one of the agonists of the beta cells, simply because the authors actually were authors of this original paper from the 90s from the Onkang group. But the problem with that original study was that they used AVP, which was in the nanomolar range, and this is not physiological range of AVP. AVP works in picomolar range, so this would be like 1,000 times lower. So if we apply the ramp of AVP, we can see that at 10 nanomolar concentration, which has been used in the original study, we are already inhibiting the activity of beta cells. So these are beta cells here, below there are alpha cells. They are also indicated where they are in the islet. AVP would specifically activate both alpha and beta cells. I will show you that they are activated in a bit different concentration ranges, but the most activity actually has been induced in the physiological range of AVP, not later on at the concentration which is typically used in the experiment. And that if you use the concentration of AVP, which is too high, also insulin release from islets would actually be diminished comparison to just to 8-glucose, so without AVP. So AVP can be stimulatory only under certain conditions. So it has to be, the cell has to be mildly activated by glucose, and then I can actually stimulate in kind of permissive potentiating manner also additional stimulation. If the beta cells are overstimulated already, then basically I will mostly inhibit the activity. And if you take a look at the literature, you will see that there is a lot of controversy regarding the AVP signaling in beta cells. So some of the papers say it's positive, some it's negative, some have no effect. And we can actually explain that, because if your cells are weakly stimulated, so all these concentrations up to maybe 100 picomolar would actually stimulate beta cells. A bit lower concentration, higher concentrations are needed for alpha cells. But if you actually go to much higher concentrations, or if you use higher glucose. So already if I use 10 glucose, practically I don't have this stimulatory activity of AVP anymore because the cells are hyperactive already. So just the glucose, 10 glucose would already so much stimulate IP3 receptors that IP3 concentration would start to inactivate IP3 receptors. And that's why we would see also this inhibition of insulin release from beta cells. So the effect, either positive, no effect, or negative effect, really depends on the functional state beta cells is from, how much glucose you stimulated them initially. Yeah, so that's a problem. To conclude, I would like to always keep in mind there are alternative targets for ATP, you know, and particularly SERCA and renin receptors are two of them that I would want to emphasize. You know, the concentration of ATP in our hands is typically very high and very constant, you know, but I would imagine that, you know, the local concentration, local, you know, kind of use can actually change that. So I think this is very important to understand, particularly if you're signaling with, you know, kind of ER, close to the ER. ER is very heterogeneous. You can have completely independent parts of ER which don't communicate each other. Everything what happens on the plasma membrane is more or less synchronized, simply because the plasma membrane is rather simple kind of like bag, and it's the electricity travels very fast there, while on the ER this is not the fact. So beta cell work very fast, and then most of the music actually comes from intracellular calcium release. I didn't show that, but if I block voltage activated calcium channels, the activity stops only about 15 minutes later or something like this. So if you can imagine that you're playing a bagpipe, you know, we can play for a while, you know, without constantly blowing into your bagpipes, but you have to feel it from time to time. And this is how I understand the role of voltage activated calcium channels, store operated calcium channels, and everything where calcium can come from the extracellular space. In beta cells, IP receptors and renin receptors really are these interactive fountains. If I block one, like IP3 receptor signaling, renin receptors will work better, simply because there's much less leak on the ER, and the pressure of calcium, let's call it like this way, can get higher, and these jets of calcium that actually stream through renin receptors can actually get higher and can coordinate the cells better in this way. And whenever we are thinking about stimulation protocols, whatever, you know, kind of like pharmacological compound we are using, it's good to understand that the same compound at different concentrations or on different backgrounds can have very different effects. And if we don't understand this multidimensionality, the interpretation of the experiments will be practically impossible. Thank you very much, and I'm looking forward to questions.
Thank you very much, Marjan. That was great. I really liked a lot of your analogies, the fountain, as well as that sort of the network of the cells, and the reference to, you know, physics was really, really interesting, and something we don't normally hear that much about, and probably should hear more about. So I have a couple questions for you too. You know, in terms of the ER, you're talking about like the different domains, right? For example, the ER right now, the primary cilia has sort of had a moment, right, in beta cell functionality, or is having a moment. And you know, I think that my understanding is primary cilia can be patch clamped, and also the ER. We talk about different domains in your talk. There's a different domain that sort of services the primary cilia. What do you think about the role of this primary cilia and patch clamping it, learning more about its calcium mechanics?
I know that the cilia has a moment. It's also good that it has. I think the cilia play, you know, we just started to see what everything cilia can do. We have been kind of like interested in cilia based on our theoretical work, where we, you know, we understand islet primarily as a sensory organ. Yeah? And as such, basically, it is critical to understand that the typical size of an islet, which is about 100 micrometers, is actually optimal to the way that basically all the cells kind of know what the other cells are doing, because they have these weak correlations which is equivalent to eyebrow movement maybe within human population. So they can very weakly feel what all the other cells are doing. But on the other hand, the size of the eyelid and the distances are already big enough for them to not be completely cross-correlated. And this theoretical work actually was kind of like suggesting there might be some movable kind of like components which can even improve, like some antennas that can improve the sensory quality, you know. Then that's why we wanted to work with the cilia many years ago already, but we did not have such beautiful tools as Jing has now in St. Louis. Regarding the
she
patch
just released
clamping,
another fantastic paper last week.
Exactly. The issue is that I think I would not. I mean, although I'm trained patch clamper, it's a very brave task to do. You know, it can be, you know, but I patched sperm in my life, but it was to prove the concept, not to actually do it routinely, because I needed like half an hour to tame, you know, the machinery of that cell. So I think it's a hard task. Hopefully, we can also find some alternatives to actually get to closer to the story, you know. But of course, you know, maybe just few limited number of patch clamp experiments could yield enough data so we could also learn what to do alternatively to study that.
Yeah.
But definitely, as you said, you know, ER and cilia are similar. They are really kind of like tubular, reticular, you know, kind of components, which don't rely that much on the conductivity, you know, because these tubes are very narrow, and the electricity doesn't travel that very well. But it actually has a lot of surface, and it can actually produce, you know, a lot of different physiological, you know, processes can run also in this kind of limited space.
Yeah, it's a really interesting time and structure. I wondered if you had any, this is sort of on the other end of the spectrum, if you have any thoughts on diet. So if you just did a thought experiment, if a human, you know, just thinking about your results, if a human's on a low carb diet, would their response have a different baseline or set point of response? I mean, because you showed you added in and took away, added in and took away the different characteristic or the different you know, the rhianodine and the acetylcholine. But I mean, if somebody's already baseline, you know, kind of lower secretion of insulin, would they have different responses in your you know, experimental setting?
I mean, what we actually did so far, partially also is a caloric restricted mice. This was the Rafael Drigo in Nashville, and the cells, they do change, but you know, we were building on the fact what also Rafael published already is that basically the cells, the longevity of cells, the cells live longer, and they don't reproduce so much in the caloric restricted islets. And we thought our original idea has been that basically, since they are like an older team, you know, they could be better, you know, they could be better in terms of their, you know, synchronous activity or something like this. But it turned out just the opposite is the case, you know. So, you know, I would, and also some other data that we have would point to the fact that the most efficient control of blood glucose actually can be achieved with IP3 receptors only, and that this synchronicity, it's not necessarily, you know, something that beta cells can do for a very long time without being killed at the end, you know. So we actually have also have some theoretical work which is published in some remote journals, which because nobody else would actually want to read that, because we wanted to address complexity within beta cells, and we compared beta cell activity to immune system. You know, immune system has kind of like some baseline activity which can be expanded to cover most of the attacks on our body. But every time when the kind of attack is stronger, we can also activate stronger mode of the immune response. But it is very unwise to actually always use maximum response, because this kills you as well as the intruder, so that's the problem. So it could be that the ranodin part is like something that gets activated in extreme conditions, where there's really overload of, and it can be used very in a limited setting. But the normal operation, particularly in human, I mean, this cephalic component is very, very strong. Like, we know that we can release a lot of insulin way before any nutrients get absorbed. We know that if we order the food and the waiters forget, we get very, very angry and nervous because of the hypoglycemia.
Oh, no.
Yeah.
Yeah.
And
Real life
therefore,
adventures.
yeah. So this is like,
Well, I mean, yeah, that's really interesting that you guys did that experiment. I'm going to look that paper up. We recently in May, we had a debate between Guy Rudder and Matt Marans and company regarding a new model for beta cell, functionality. And then recently, Matt Marans and Richard Benninger came out with a paper, a bioarchive preprint
now I
glucokinase.
remember these alternating cells.
Yeah,
Yeah,
control
right,
cell
right.
populations of beta cells that alternatively lead islet calcium oscillations. I didn't know if you wanted to sort of weigh in on that as much as you'd like to.
No, I mean, regarding, I was very happy that basically, that metabolism actually came back to this kind of central stage, and that it has a very kind of like very nice discussion on your platform too. It was very well visited or something. But I think that one of the limitations, I also discussed that with Matthew, is that the focusing on just on the KTP channels might be a bit of a problem for me. So I think it is, we have tons of ideas we follow simply because there is evidence that actually shows that even why this discussion was very important to me. It also what Ian Sweet was actually showing us, these different points where glucose can produce ATP. It's either glycolysis, it's TCA, or it's even post-TCA, kind of complex one, a generation of ATP. And this is something what, it was not, as a physiologist, I was not really thinking along these terms. So we immediately set up the glyceraldehyde experiment, which we never did before. And to our surprise, glyceraldehyde is very, very strong stimulation, which you can find in literature. It's just that we don't really understand why it is so efficient, and it's in our hands. It is primarily stimulating very coordinated activity between beta cells, suggesting that this kind of complex one activation likely communicates with renin receptor more efficiently than with IPT receptors. So there could be also distinctions which of part of the metabolism is actually controlling ATP production for different calcium release. Particularly, we always forget about the circuits, which actually have to bring back calcium into the ER. And I hope we particularly, in the Indianapolis group actually has models that actually can address that. And we'll be happy to see that, how it works.
It seems like it's a time for electrophysiology, patch clampers, physiologists, metabolists, if you want to call them metabolists, to all get together and put their heads together on this very complex question, and hopefully push the ball forward. It looks like you're at least thinking in that direction. So thanks for sharing all this great data, and also your great interpretations of it. Appreciate it very much.
Thank you very much for inviting me again. Yeah.
All right. Have a great rest of the day, Marjan.
You too.
Thank you.